Hello, everyone, and thank you for joining today's webinar, Adding Hepatic Organoids to Your Research, presented by Dr. Charis Segeritz-Walko. After earning her master's degree at the University of British Columbia in 2011, Dr. Segeritz-Walko went on to complete her PhD at the University of Cambridge under Dr. Ludovic Vallier. During this time, Dr. Segeritz-Walko investigated the differentiation of endoderm-derived tissues from pluripotent stem cell, particularly of the hepatic lineages. After completing her PhD in 2015, Dr. Segeritz-Walko joined STEMCELL Technologies' research and development team working on the development of organoid growth media for epithelial tissues. She has since led the development of hepatic and pancreatic products, recently releasing hepatoccult and pancreacult organoid growth media. Thank you so much, Charis, for agreeing to speak with us today and for sharing your knowledge on culturing hepatic organoids.
Thanks so much for the introduction. It's a pleasure for me to talk to you today about liver organoids and the applications this culture system has found in the hepatic research field. As Nick mentioned, my own background is in the differentiation of human pluripotent stem cells to hepatocyte-like cells. And so the emergence of a three-dimensional culture system that still allows the study of hepatocytes in a patient-specific background has, in my eyes, certainly revolutionized the research field. Today, I would like to introduce you to liver organoids, their biology and physiology, and to outline their derivation in the context of the liver-residing stem cell niche. Next, I will discuss some examples of applications for liver organoids by presenting some of the most groundbreaking studies that have been published to date. I will give insight into a few common challenges and obstacles that can be faced when culturing hepatic organoids. And finally, I would like to present some practical advice on how to get started with liver organoids and how to harness them in addressing your specific research questions. At the end of my presentation, I'm happy to address any questions you may have, but feel free to send them in at any point during my presentation. So let me start us off by introducing you to liver organoids. In order to appreciate the long way we've come in the liver organoid research field, we need to go back 5 years to the first publication that has emerged on liver organoids. Marisa Hook, at the time working in Hans Clever's lab, and her colleague showed in this Nature article how liver organoids can be established from mouse liver tissue. This study, of course, ignited the question of whether hepatic tissue could also be propagated ex vivo from human liver tissue. So the follow-up study from the same group showed that liver organoids can indeed be established from human liver biopsies. It also demonstrated that organoids derived from liver biopsies of patients with inherited liver disorders do preserve the pathophysiological disease signature. This finding confirmed the enormous disease modeling potential of organoids, including the dissection of molecular pathways that underlie disease and the development of new drug therapies targeting these disorders. And finally, last year, the application of human liver organoids was extended to also model primary liver cancer. The tumor-derived organoids displayed the phenotype of the parental carcinoma and were utilized to identify cancer-specific biomarkers and drug targets. These findings established the utility of organoids not only in the development of drug therapies, but also their use as patient avatars that can inform personalized cancer treatments. So we see the progression where the first proof of concept studies using mouse tissue have quickly evolved into highly relevant tools that address both basic and translational research questions. In the near term, organoids are definitely showing huge promise for drug discovery pipelines and clinical applications in the area of diagnostics. But of course, the long-term goal is the incorporation of organoids in personalized cell therapy. So before we dive further into the exciting applications of liver organoids, I would like to take one step back and touch base on the underlying liver biology that allows us to establish liver organoids in the first place. The liver is composed primarily of hepatocytes, which are shown here in pink. Hepatocytes are the metabolic workhorse of the liver and carry out the specialized tasks of this organ. This includes the synthesis and storage of amino acids, proteins, vitamins, and fats, blood glucose regulation, bile drainage, and the circulation, filtration, and detoxification of blood. Hepatocytes are epithelial cells with a lifespan of approximately 5 months under homeostasis, so this is a very slow turnover. For example, in contrast, epithelial cells that line the small intestine are renewed on a weekly basis. In health, hepatic cell mass is retained, although the source of renewing cells has been widely debated. Adult hepatic stem cells have been postulated to reside in the canal of Hering, which is also known as the intrahepatic bile ductules, and these are partially lined by cholangiocytes, bile duct cells, which are shown here in red, and hepatocytes, shown here in blue. This renewal is hypothesized to be driven by axin 2, which plays an important role in the Wnt signaling pathway. In injury, damage-induced LGO5 positive cells have been shown to generate hepatocytes and cholangiocytes in vivo using lineage tracing studies, but LGO5 is not expressed in the healthy liver, which suggests that in the liver, different cells or genes are responsible for regulating the processes of homeostasis, acute and chronic injury. Hepatic progenitor organoids are therefore derived by isolating the liver ducts and culturing the liver stem cells within them. Next, I would like to give a brief overview of the experimental procedures to establish, maintain, and differentiate liver organoids. The concept is comparable between organoids derived from mouse and human liver tissue and starts off with dissecting out the liver of a mouse or obtaining a small human liver biopsy. The tissue is minced and then exposed to enzymatic digestion to free up the intrahepatic bile ducts that contain the liver stem cell niche. These ducts can either be collected whole or digested further into single cells. Ducts are isolated from the enzymatic digest through hand picking, or our lab has also developed a filtration protocol to enrich for these ducts, and it's a much easier and less labor-intensive method than manual selection of the right material. Single cells can be enriched further by FACS using stem cell markers LGF5 or APCAM, or we have also established that organoids grow when plating the whole bulk single cells without sorting them first. Regardless of the isolation procedure, the biological material is then mixed with an extracellular matrix, such as matrigel. This provides cells with a three-dimensional scaffold to grow into organoids. This method has also been postulated to be more physiologically relevant than two-dimensional monolayer cultures, as it better mimics the physical signaling the cells are subject to in vivo. Besides the addition of matrix, the biological material is also mixed with an organoid expansion medium that mimics the chemical signaling of the hepatic stem cell niche. When embedding the ducts or single cells in 100% matrigel and plating them as domes, the extracellular matrix gels and the wells can then be flooded with the organoid expansion medium. Alternatively, the isolated ducts or single cells can be mixed into a suspension culture containing only 10% matrigel diluted in the organoid expansion medium. We have developed this suspension culture as a method to quickly generate a large liver organoid biomass, since plating of these suspension cultures is easier to adapt to a high throughput workflow and also doesn't require medium changes between passages. After embedding the primary tissue material in dome or suspension culture, the cells self-organize within a week into epithelial monolayer spheres with a hollow lumen. These hepatic progenitor organoids are composed of cells that still express liver progenitor and stem cell genes. At this stage, organoids are genetically stable. They can be maintained indefinitely by passaging the culture, and within a week, these passaged organoid fragments luminize and expand again into new organoids. Established organoids can also be cryopreserved for long-term storage and cultured as organoids again at a later point in time. As I mentioned before, hepatic progenitor organoids have an active stem and progenitor cell population, which allows indefinite culture of hepatic progenitor organoids. But this comes at the expense of mature hepatocyte gene expression, and hepatic progenitor organoids do not yet exhibit significant cytochrome P450 or other liver-specific detoxification properties. So in order to mature these progenitor organoids and to obtain cells that display hepatic function, Passage organoids are transitioned from the expansion medium after 2 days to a differentiation medium. The composition of the differentiation medium stops Wnt and TGF-beta signaling and allows for mature liver genes to be activated. Organoids are formed within 2 weeks and display a thickening of the epithelium. Once they're differentiated, organoids cannot be passaged or cryopreserved since the stem cell population is lost in the process of differentiation. For those of you who are familiar with the mouse intestinal organoid system, you will notice an expansion and differentiation step is required to obtain differentiated cell types in hepatic organoids, while mouse intestinal organoids are obtained with a single culture medium. This is reflective of the intestinal versus hepatic physiology, since the intestinal lining in the body is composed of both differentiated and highly proliferative cells at homeostasis. The signals for proliferation in the liver, however, are typically quiescent in regions of mature hepatocyte function. So far, we've established that liver organoids are obtained through isolation and expansion of stem and progenitor cells from hepatic stem cell niches, that different culture formats are amenable to maintaining and expanding organoids, and that hepatic progenitor organoids need to be differentiated further to obtain a mature hepatocyte phenotype in vitro. So let us next explore the various applications of liver organoids in the hepatic research field and why the organoid culture system has in fact been termed Method of the Year 2017 by an editorial in the Nature Methods published last month. And to me, this is just one example of how the liver and research field at large is recognizing the great potential of the organoid culture system. Cultures that rely on tissue-specific adult stem cells have enormous potential to expand the breadth of hepatic research being done in quite a variety of labs, and that's just by virtue of the fact that they present a significant advantage over some of the traditional culture methods, for example, primary hepatocytes or immortalized cell lines. And we'll go into these into greater detail in the following slides. In order to highlight the various avenues of hepatic research that have introduced and incorporated liver organoids so far, I would like to go back to the hallmark papers I introduced you to at the beginning of my presentation and show you examples of organoid use in both mouse and human cell models. To start off with, the proof of concept paper from Hook et al. In 2013 thoroughly characterized the organoids and showed that these liver organoids preserve key physiological features of the liver. In so doing, this paper shed light on the potential applications of liver organoids. Because these cultures exhibit key features of the liver epithelium, they can be enormously useful as an in vitro liver model in a wide variety of research contexts, including studying liver biology and modeling liver diseases. Going back to the actual protocol used, liver damage was induced in mice to upregulate the injury-responsive stem cell population marked by LGR5. Single-cell-derived organoids emerged with the expression of stem and progenitor markers LGR5, SOX9, and cytokine 19, and mature markers such as albumin, FAH, and CYP were not expressed at this stage yet. But upon introduction of a differentiation medium, the stem and progenitor markers were downregulated while mature liver markers were upregulated. These differentiated organoids were transplanted into mutant mice suffering from acute liver failure, and organoids engrafted into the tissue partially rescued the mutant phenotype. This publication showed that organoids can emerge from both whole biliary ducts and also from single cells isolated from mouse livers. The organoids consist of a spherical monolayer epithelium, and the organoids can also be passaged long-term. This characteristic represents a huge advantage compared to other in vitro experiments that are based, for example, on primary hepatocytes, since organoids allow for continuous supply of hepatic cells without needing to go back to an animal for tissue. To understand the cellular identity of hepatic progenitor organoids cultured in expansion medium, Hook et al. Compared the gene expression profile of 2 liver organoid cultures to different tissue types, including the liver. As you can see from this heat map, both liver organoid progenitor cultures clustered more closely with the expression profile of the liver than the other tissue types. Furthermore, when looking at specific duct and hepatocyte genes, organoids cultures displayed gene expressions that were in agreement with both cell types, suggesting a bipotent nature of these hepatic progenitor organoids. This was further solidified by comparing marker expression of hepatic progenitor organoids, marked here as culture 1 and 2, against adult liver tissue. Stem and progenitor markers were expressed in organoids, but not in homeostatic liver tissue, confirming the proliferative nature of hepatic progenitor organoids versus the quiescent nature of liver tissue. They also characterized hepatic organoids cultured in differentiation medium by looking at the synthesis of mature hepatic proteins. The publication reported positive staining for the serum protein albumin, the transcription factor 18F4-alpha that drives expression of many mature liver genes, the multi-drug resistance protein MRP4, and the cell-cell junction marker Z1. But you can also see that some cells synthesize the ductal marker cytokinin 19, and that EpCAM, the surface marker of hepatic stem and progenitor cells, was also still present. This indicated that the differentiated hepatic organoids were not fully mature and also retained a population of ductal cells. However, there was increased hepatocyte function in organoids exposed to differentiation medium, annotated here as DM, and there was an increased hepatocyte function in organoids exposed to the differentiation medium, annotated here as DM, and as seen by the ability of cells to uptake low-density lipoproteins, shown in the right picture here in red. Differentiated organoids also showed intracellular glycogen storage as detected by PAS staining. To capture the detoxification properties of the cells, the authors then compared the enzymatic cytochrome P450 activity of organoids in expansion medium, EM, and differentiation medium, annotated as DM, to a negative control, MEFS, and also a commonly used alternative to liver organoids, the transformed cell line HepG2, as well as primary hepatocytes as positive controls. You can see that there was a 7-fold increase in enzyme activity when transitioning organoids from the expansion to the differentiation medium, and significantly, this activity was higher than that seen in HepG2 cells. The hepatic subactivity of differentiated organoids was still 24-fold lower than what we would have anticipated in primary hepatocytes, but nonetheless, this represented a significant advantage over the commonly used cell line HepG2. These observations also rang true when the authors measured the levels of albumin secretion. The secretion of albumin significantly increased as organoids were transitioned from expansion to differentiation medium, but again fell short of the capacity displayed by primary hepatocytes. In the light of this lag in maturity seen by hepatic organoids in vitro, the authors next wondered whether hepatic organoids could engraft and further mature when transplanted in vivo. To address this question, the authors transplanted differentiated organoids or controlled primary hepatocytes into FAH-deficient mice. Without the enzymatic activity of FAH, these mice are subject to liver failure. After 2 to 3 months post-transplantation, only mice that had received differentiated organoids or primary hepatocytes showed positive staining for FAH, indicating that differentiated organoids had endogenously contributed to the synthesis of FAH. Grafted cells were also found negative for the ductal marker cytokinin 19 and positive for the hepatocyte marker 18F4-alpha, suggesting that these cells had indeed matured to a hepatocyte-like phenotype in vivo. Overall, when mapping out a Kaplan-Meier survival curve of transplanted mice with positive or negative engraftment, the authors reported a significant increase in survival of the positive engraftment group compared to the graft-negative mice, confirming that the transplanted cells had contributed to liver function in vivo. As a proof of concept study, this publication created a lot of excitement in the research community and led to the question about possible implications for studying the human liver in health and disease. Since human tissue is significantly harder to come by than primary mouse tissue, the value of a human liver organoid system is even greater. It would lessen or even remove the restriction of liver biopsy availability and expand the breadth of studies a given lab would be able to perform. So to transition between species, the protocols and medium formulations to grow liver organoids had to be optimized. But indeed, Hook et al. Showed in 2015 that it is also possible to generate liver progenitor organoids and differentiated organoids from human liver biopsies. Similar to the mouse organoid system, organoids downregulate stem cell genes when transitioned from expansion to differentiation medium, while upregulating in parallel markers associated with mature hepatocyte and ductal lineages. This was also evident on a functional level when comparing the CYP3A4 activity and bile acid production between organoids cultured in expansion and differentiation medium, EM and DM, and also in comparison to a negative control, feeder cells or HepG2 cells. CYP3A4 activity and levels of bile acid synthesis were the highest in organoids exposed to differentiation medium. In both cases, the organoids cultured in the differentiation medium represented a significant advantage as an in vitro model system over the HepG2 cells. Differentiated organoids also showed protein synthesis of the mature liver markers we have previously discussed. And when transplanted into mice, human albumin was detected in the graft, confirming the possibility to transplant and further mature organoids in vivo. Upon thorough characterization of these organoids, the authors then moved on to demonstrate that liver organoids can be used to model inherited liver disorders. Establishing an in vitro culture system that mimics disease in a dish allows us to dissect the molecular pathways that give rise to the disease. It also allows us to use them as an in vitro liver model during pharmaceutical compound discovery. But maybe most importantly, this patient-specific disease in a dish paves the way for precision medicine and development of tailored drug therapies. An example studied in this publication was the genetic liver disease alpha-1 antitrypsin deficiency, which leads to liver fibrosis and cirrhosis in vivo. The differentiated organoids derived from the liver biopsy of an alpha-1 antitrypsin deficient patient were seen to exhibit many key characteristics observed also in patient tissue, including intracellular alpha-1 antitrypsin protein aggregates, protein secretion patterns, ER stress, and apoptosis. Another genetic disorder affecting the liver, Alagille syndrome, was also modeled in this paper using patient-derived liver organoids. These characterizations were an important proof of concept that the liver organoid model system has great research potential for modeling key disease phenotypes in vitro. Overall, the groundbreaking aspect of this publication was that within 3 to 4 weeks, inherited liver disorders were phenocopied in patient-derived organoids. Previously, studies had to rely on first the reprogramming of a patient's somatic cell into induced pluripotent stem cells, and then their subsequent differentiation to hepatocyte-like cells, an undertaking that's a matter of months rather than weeks. So when it comes to swiftly diagnosing a disease and developing a personalized treatment plan, time is definitely key, and liver organoids seem to be the more suited platform. Additionally, the use of adult stem cell-derived tissue may keep the derived cells closer to the organ's tissue biology than human-induced pluripotent stem cells, and also reduce the risk of errors in imperfect directed differentiations. The last publication for human liver organoids I would like to discuss today is based on last year's publication on human liver tumor organoids, also known as tumoroids. Primary liver cancer typically manifests as hepatocellular carcinoma, annotated here as HCC, cholangiocarcinoma, annotated as CC, or as a mixed tumor identity, annotated as CHC. To study these different tumor subtypes, the authors used H&E staining of healthy and tumorigenic resected liver tissue, and in parallel established organoids from these tissues. While healthy-derived liver organoids grew with a single-layer epithelium surrounding a central lumen, as seen on the left, tumor-derived organoids formed compacted structures that resembled the corresponding tumor of origin. Based on RNA-seq data, the authors then compared several tumor tissues with organoids derived from that tissue. The labels were annotated as ending with T for tissue or O for the corresponding organoid cultures. They found that the genomic landscape of the derived organoids identified the parental tumor subtype and retained mutations which were patient-specific and therefore could be used as prognostic markers. The gene expression profile of the tumoroids was also indicative of the proliferative or differentiative status of the parental tumor. And as if this wasn't striking enough already, the authors then transplanted tumoroids under the skin of immunocompromised mice to determine whether in vivo tumoroids also reproduce the features of a human primary liver tumor. And indeed, tumor outgrowth were found only in the animals engrafted with tumoroids, but not when injected with healthy liver-derived organoids. Since primary liver cancer metastasizes primarily to the lung and also the lymph nodes, tumoroids from a patient with a history of metastasis were transplanted into immunodeficient mice. As expected, 100% of the injected mice developed tumors that resembled the original patient tissue, but more excitingly, in 7 of the 9 injected mice, the authors found lung metastasis in agreement with the patient's diagnostic at the moment of resection. To take the study even one step further, the authors carried out proof of concept drug sensitivity testing in 6 primary liver tumoroid lines. The goal was to evaluate their capacity to identify patient-specific drug sensitivities and to assess the utility of these tumoroids as a platform to inform drug development and drug administration. The authors tested the organoids' sensitivity to 29 anti-cancer compounds, including drugs in clinical use or in development. The tumoroids were treated with a dilution series of each compound, and I'm showing here the example of the ERK inhibitor SCH-772984. In the 10 micromolar range, several tumor lines showed reduced viability. This was also evident in the morphology of the tumoroids treated with this ERK inhibitor. Pictures shown in red boxes indicated no viable cells and therefore sensitivity to the drug, while the non-box pictures showed organoid cultures that were resistant to treatment. To test the efficacy of this compound at inhibiting tumor growth in vivo, tumoroids were transplanted into immunodeficient mice, and when tumors had reached 100 cubic millimeters in size, the ERK inhibitor or a negative control were injected into the tumor. 2 to 7 days after the first injection, the authors observed significant reduction in tumor growth, which lasted for the remainder of the experiment. Histological analysis revealed that the tumor mass was necrotic and that the majority of the cells were apoptotic. So this has shown us that a liver tumoroid system can be a good platform to screen and identify an anti-cancer compound at the correct dose that this patient, or in this case a mouse, would be sensitive to and leading to a shrinkage of his or her tumor. So again, this is a huge leap in the direction of personalized medicine, and I'm definitely excited to see how these liver organoids and tumoroids will be incorporated into the medical healthcare system in the coming years. So to summarize these last slides, human liver organoids can be utilized to study liver cell biology and regeneration. We know that the liver can regrow when removing more than half of its mass, so powerful mechanisms of regeneration must be at play, and liver organoids can be an interesting model to study the role of hepatic stem cells in regeneration. The characterization of human liver organoids we just looked at also indicates that many of the liver-specific genes, protein synthesis profiles, and functional characteristics are preserved in organoids, and this makes them a physiologically relevant cell culture system for studying basic liver cell biology. Liver organoids can also be used to model inherited liver diseases, and we've seen this in today's webinar with the examples of alpha-1 antitrypsin deficiency, and I touched on Alagille syndrome. Liver organoids can find employment in modeling hepatic cancer, and the data I discussed showed that liver tumoroids faithfully model the genetic complexity of human primary liver cancer, and that they can be used to develop individualized drug treatment strategies. And lastly, something we haven't touched upon today is the use of liver organoids in liver toxicity assays. By using markers of apoptosis, liver organoids can be used as surrogates for testing the toxicity of novel drug compounds. And similarly, I would like to highlight the applications in which liver organoids can be advantageous over other liver models. So first of all, primary hepatocytes. They are still the gold standard when it comes to evaluating hepatic metabolism and toxicity of drugs or other xenobiotics in vitro. However, for many other applications, liver organoids are much longer lived and also less challenging to source. Immortalized cell lines such as HepG2 cells are convenient to culture in 2D, but they are genetically abnormal and therefore in some applications questionable in their physiological relevance. Animal in vivo models are time-consuming and costly to incorporate into liver research, and the whole animal model also can include many confounding variables compared with isolating the tissue of interest. But I can see also room for complementary work here. For example, gene editing techniques can be carried out in liver organoids much faster and in a more controlled environment, and can then be verified in vivo animal studies. And lastly, human biopsies are non-viable and therefore difficult to interrogate for specific mechanistic studies, whereas organoids provide an in vitro system where individual experimental variables can be easily manipulated and thereby greatly expanding the options for experimental design. So I hope I've demonstrated to you in this section that organoids consist of a spherical monolayer epithelium that preserves key physiological features of the liver, and that this culture system can be used to study hepatic development and regeneration, inherited liver diseases, primary liver cancer, and that these organoids carry promising applications for cell therapy as well as in vitro diagnostics. You might be asking, given these diverse applications for hepatic culture systems, which faithfully model liver biology in vitro, why isn't everybody using this technology? In this section, I would like to talk to you about some of the common challenges that researchers may face when using the organoid technology in their labs. It is a very recent technology, and while it holds enormous research potential, it will also take some time for widespread adoption. But I would like to draw attention back to the Nature Methods editorial, which pointed out that the ability to prod stem cells into three-dimensional tissue models makes for a powerful way to study human biology. But these exciting tools are still works in progress. As with any new technology, currently there are temporary challenges to incorporating organoids into the liver research field at large. While many characterizations have already been carried out on liver organoids, there are still unanswered questions and clear gaps in this technology. We, for example, still see room in improving the existing protocols and formulations to enhance the functional repertoire of liver organoids. But we can see the academic and industrial research field is actively working together on overcoming these obstacles and streamlining the processes. So I would like to briefly outline these challenges that are currently being tackled, both in our lab and in others. So first of all, without the starting material, of course, there won't be an organoid culture. And although it is handy that once organoids are established, they can be cultured indefinitely and don't rely on further tissue supply, we have to start with some tissue. For culturing most organoids, this is easily overcome, as all that is needed is access to an animal facility or the purchase of commercially available mouse liver organoids. For culturing human organoids, however, access to a biobank or collaboration with a hospital is typically required. It's important to initiate organoid cultures from high-quality tissue, preferably within less than 24 hours after resection. If tissue cannot be obtained, sometimes cryopreserved organoids can be requested from labs that do have access to primary tissue, but this process is typically more straightforward in the academic sphere than for biotechnology companies that are looking to start an entire organoid culture program. It goes without saying that accessing and working with murine and adult human tissue is regulated and needs to be approved by local jurisdictions and ethics review boards. Next, all organoid expansion media are reliant on high levels of growth factors, which are costly if purchased as recombinant proteins. So many of the organoid medium recipes rely on the use of transformed cell lines that secrete growth factors into the medium to make what's called conditioned medium. Although this can significantly reduce material costs, it is timely to set up cell lines to continuously generate conditioned medium, and it can easily become a strain on lab labor and space. But maybe more importantly, it is highly challenging to consistently produce highly controlled conditioned medium that contains the same concentration of growth factors in every batch. So if this is not achieved, this sometimes introduces variability in the growth of organoids. As a side note, the liver differentiation medium, in comparison to the expansion medium, does not rely on the use of conditioned medium and contains fewer factors and is therefore relatively straightforward to prepare. As we've discussed in this webinar, organoid growth is reliant on extracellular matrix, and matrigel is commonly used for that purpose. However, it's also an undefined material that is subject to significant variability. Matrigel is also a tricky substance to handle and work with. It is reversibly solid at -20 degrees, liquid at 4 degrees, and irreversibly solid at 37 degrees. And also, speaking of matrices in general, they are currently not conducive to high throughput methods and scale up. In order to address these challenges, for example, we have introduced the use of the suspension culture system. And I will go into that into more detail in my last slides. And as a final note on matrices, when it comes to objects growing in 3 dimensions and in different planes, there's also an additional complexity that is added when it comes to imaging these objects. And the last point I would like to touch upon is the lack of functionality displayed by differentiated hepatic organoids when compared to, for example, primary hepatocytes. This is currently still a shortcoming in all in vitro hepatocyte cultures, so it's important to keep in mind what the applications of our liver organoids are and whether our organoids are up to scratch to meet these demands. For example, mimicking complex genetic disorders of the liver, yes, certainly organoids can do that, as we've seen today. But predicting the rate of metabolism by the liver with respect to a novel drug compound, we're probably not there yet. So now to discuss some of the work that still remains to be done to make organoids even more accessible, I would like to finish this webinar by sharing some practical advice on how you can already take advantage of liver organoids by utilizing the available resources. So let's start off with the incorporation of human liver organoids into your research. As discussed earlier, an important part is the access to biological starting material. The most frequent route is via hospitals and biobanks. And we've seen great collaborations and translational research emerge between academics and the local hospital. And so these relationships with surgeons who work or are interested in this intersection of clinical work and academic research are great partners to pursue. An outstanding resource for detailed protocols on establishing, maintaining, and differentiating liver organoids from mouse and human tissue is this Nature Protocols publication. It also contains recipes for the organoid media and includes step-by-step instructions on how to produce conditioned medium. The article also contains protocols for genetically manipulating the generated organoids, for example, by using transfection and transductions. For mouse liver organoids, we have recently developed hepatic organoid growth medium for the initiation and maintenance of hepatic progenitor organoids cultured from mouse tissue. It's a complete defined medium that consists of a supplement that is simply added to the basal medium. This is a great simplification over the published formulation that relies on a cocktail of 15 components and the preparation of conditioned medium. Hepatic is supplied with straightforward protocols that are flexible to be tailored to the priorities of your experiments, and STEMCELL Technologies also provides technical support through our product and scientific support department, who are available by phone or email to address any challenges that may arise. For labs that don't have access to animal facilities or are looking to eliminate animal use within the experimental workflow, or just want to initiate their mouse organoids as quickly as possible, we also offer cryopreserved mouse hepatic organoids, which can be expanded indefinitely using hepatic. These organoids are derived from the liver of a wild-type Black6 mouse. Hepatic supports the growth of isolated whole hepatic ducts, hepatic duct fragments, and also single cells. Once organoids are established, either through primary culture or the thawing of cryopreserved organoids, they can be passaged indefinitely without any detrimental effects on their genome stability, gene expression, or morphology. Established organoids are typically split at 1:25 ratios, and when mapping cumulative cell yield across 10 passages, we observe exponential growth in organoids cultured with hepatic cult, allowing for rapid expansion of the hepatic cell population. When we closely investigate the morphology of the hepatic epithelium of these organoids, we see cells displaying the typical polygonal architecture of the liver epithelium, and also binucleation in some cells, another common liver feature. RNA-seq of the hepatic organoids compared to organoids cultured in the published expansion medium showed that these cells clustered very closely to their published counterparts in this PCA plot. We also confirmed the appropriate gene expression and capacity for differentiation using the published Hook methodology that I reviewed earlier in this presentation. And if you're interested in more information and data on the performance of this medium, you can visit our website at hepaticult.com. As my last piece of data, this video allows you to follow the growth of hepatic progenitor organoids over a period of 5 days. Hepatic progenitor fragments at passage 7 were seeded into matrixal domes, cultured in hepatic cult, and then imaged in an incucyte zoom every 2 hours. You will notice that the organoids start exhibiting the typical cobblestone morphology towards the end of the expansion, which is typical of the hepatic epithelium. Before we finish off, I would like to discuss 3 pieces of advice that are helpful to keep in mind when culturing liver organoids, especially if you're new to this technique. The first one is pertinent to the seeding density. The matricial dome is limited in the material it can hold, and it is therefore important to optimize the material that is embedded in a given matrix volume. For example, for weekly passaging, we recommend the embedding of 200 passage organoid fragments that contain about 5 to 10 cells each in a 30 microliter matricial dome. Packing too much material into a matricial dome can decrease the stability of the matrix, and it may collapse during the culturing process. Overseeding the cultures will also result in organoids growing faster and requiring earlier passaging. On the flip side, since organoids like to grow in company of others, underseeding the cultures will result in reduced organoid growth rates. Especially when getting started with growing organoids, it's a good idea to monitor organoids daily to get a sense of how quickly the organoids grow and need to be passaged. Speaking of passaging, it's oftentimes a solution if something doesn't go as expected in the culturing process. For example, if no organoids emerge in the primary culture, if matricial domes collapse or the organoid cultures appear overgrown, gathering the material and splitting it sometimes results in organoid emergence in the subsequent passage. And liver organoids are really robust, and once a single healthy organoid is established, it will readily give rise to a large number of progeny organoids upon passaging. In regards to setting up cultures based on single cells, most single cell dissociation enzymes are suitable, and we recommend the seeding of 10,000 to 15,000 cells in a 30 micron matricial dome. We also recommend the addition of ROCK inhibitor for the single cell cultures. And lastly, suspension culture. It's a very easy culture system for scaling up organoids. The material that would be embedded in a dome culture in a 100% matricial dome is instead mixed into a cooled vessel or well that contains 10% matrigel diluted in cold hepatic cult. We then transfer these cooled cultures to a shaker in a 37-degree incubator, where under rotation, the matrigel slowly solidifies. It forms a thick extracellular web throughout the culture that catches the suspended organoid fragments or single cells, and organoids emerge within a week or oftentimes earlier. The speed of rotation needs to be adjusted to the size of the vessel, but we recommend 80 RPM for organoids cultured in 12-well plates. So in today's webinar, I've shown that hepatic organoids are functional, 3-dimensional in vitro models of the liver. They consist of a spherical monolayer epithelium, which preserves the biology of the tissue it is derived from. Hepatic organoids are obtained through isolation and expansion of stem and progenitor cells from hepatic stem cell niches. I've also discussed data that has shown the application of hepatic organoids as a novel cell culture technology that enables researchers to look into the hepatic development and regeneration of the liver, metabolism studies, and liver disease modeling, including primary liver cancer. We've now reached the end of our webinar, and I would like to thank you for your attention and interest in liver organoids. I will now have a look at the questions we have received and address these in the time we have left.
Thank you so much, Charis, for taking the time to talk with us today. I'm really excited to see the potential of organoid technology and what it means for the various research fields that they're being applied. So one that we have here asks, in the Hook paper, they characterized the organoids and found characteristics typical of both hepatic and ductal cells. Were these distinct cell populations, or did some cells display markers of both cell types?
The paper only analyzed the gene expression on an organoid's bulk level. The publication would have required single-cell RNA-seq to give more insight into this question, but from the immunocytochemistry stainings that we looked at of organoids, We could see that certain populations within a given organoids were, for example, positive for cytokeratin 19, indicating that these were exclusively of ductal origin, versus the rest was mostly of hepatocyte origin. So there seems to be a mixed origin of cells within a given organoid.
Is hepatocult similar to either the differentiation medium or the expansion medium that you talked about?
Yeah, hepatic cult is similar to the expansion medium described in the publication by Hook et al. From the 2013 Nature paper. And that's in that it can be used to both establish liver progenitor organoids from primary liver tissue and maintain these organoids through weekly passaging. The gene expression profile of liver organoids that are grown in hepatic cult is very closely aligned with those cultured in the published expansion medium, as I've shown through the PCA plot. In regards to the differentiation medium, we recommend customers to transition their hepatic cult cultures to the published differentiation medium for that step.
And is there any option right now for human organoids, or is it just the published medium?
So currently we don't have a medium that can be used for culturing human organoids, and this is something that we are actively working on at the moment.
So when you're culturing organoids, what is the recommended volume of media and cells to Matrigel for dome cultures? We've had a few people ask that. It seems they're starting their own cultures.
Yeah, so usually this needs to be, the medium volume needs to be adjusted to the well format. So the recommendations that we have is that for a 30 microliter Matrigel dome seeded into a 24 well plate, we usually use volumes of 750 microliters of expansion medium, and we change the medium every 2 to 3 days. So that allows scientists to have weekends without medium changes once the medium is changed on a Friday. And that usually allows for 3 media changes before organoids are passaged.
You talked a little bit about the suspension culture and the benefits that it provides. Why is suspension culture potentially better for larger scale experiments?
I think it saves time since the Matrigel doesn't need to be used for individual dome formation. It also eliminates the need for medium changes, and organoids can be grown up in larger culture vessels, for example, in spindle flasks. It's less finicky of a technique, given that everything is basically handled in bulk.
Is there a specific protein concentration or any other aspects of the Matrigel that you recommend for growing organoid cultures?
We've seen that there has been maybe variable performance of Matrigel at lower protein concentrations. So that would be if the Matrigel concentration is below 8 mg per mil. But typically, we don't see any issues with the stability of the dome when it comes to that.
So we talk about organoids as being kind of heterogeneous collections of cells that grow up from a stem cell source. Given that the kind of stochastic nature of them, how reproducible is organoid generation from the same source? So how similar are two batches of organoids generated from the same starting material?
Yeah, that is a good question, and we do see variability in the gene expression profile between different cultures. We usually set up a couple of repeats to ensure that there is a robust readout at the end of the differentiation. But yeah, you're right in that the organoids that do emerge are not homogeneous in their subpopulation and hence not in their gene expression either. But that, I think, just highlights the need to control pretty much any other factor, including the medium and the extracellular matrix that these cells are exposed to, so that the only variable that is present is basically the starting material that is embedded in organoid, in the extracellular matrix.
All of the techniques that you've talked about so far use Matrigel. What does this mean for the potential of liver organoids for cell therapy or other applications?
I think it means that we still need to find and develop a matrix that is defined, that is reproducible and amenable for high throughput methods, and that can also be used then in conjunction with cell therapy. And that is still a work in progress. There are lots of defined matrices currently in development, and I guess they're being tested for the application of organoid growth as well. But so far, a clear winner that could perform to the levels of Medigel is still pending, in my knowledge.
We talked a little bit about the ethics of using these cultures. What would be the kind of most important consideration for the use of human organoids for people that are looking to add these to their research?
So generally, when working with human biopsies, there needs to be ethics review boards that reviews the processes, and that is something that needs to be done ahead of time. It's definitely a process that can take a few months. So if you're thinking about working with human liver biopsies and deriving your organoids, that's something to think about when getting started as soon as possible.
Great. Well, I'd like to thank you once again for taking the time to speak with us today. I found this very informative, and I hope everyone else did as well. Anyone who asked a question that we didn't have time to get to, we will try and follow up by email as soon as we can. So thank you all for joining us, and thank you, Charis, again for this wonderful presentation.
Thank you. It's a pleasure.